Okay, so it's office hours for lecture 1, lecture 2, basically gonna go around the room. Any questions you have, you feel free to say your name, introduce yourself. But if you don't want to say your name, I know this is being recorded, that's also perfectly fine. But the more I get to know you, the better. And then if you become those people who want to LA or need letters of recommendation, like this is a really good format for you to get to know me and I get to know you. Okay, let's get it started.
Okay, well, I'm not sure if this is more like for my own understanding.
That's fair, yeah.
It was like the mold experiment thing with the
The
oxygen.
oxytrophs.
It was like the diagram that's on page 46. I just want to make sure that I was like understanding it correctly
Okay.
because it took me like a while because I was looking at this and I couldn't really tell what I was looking at. For the
Okay, so let's actually just break down the types of figures that they use. So there's different ways that you're actually gonna see like depicted. So they used to use, right? They used to use. Are you seriously? Oh, oh, my spring. Okay, this is ooh, pink. Okay, so they used to use what we call a media that wasn't solid. Or it's like semi-solid. They grew things in test tubes, and then what happened is that the mold, the spores, they grew on top of the layer of media. And so that's what you see in the figures, that they're using literal test tubes. The way you normally see it displayed now, people grow things, they grow in petri dishes. So instead, you have a big flat surface that you're used to seeing, and then you look for the growth or like colonies on that plate. So that's what you're looking at in the way the diagram is drawn. And I know it looks kind of weird if you haven't seen like mold growth like that, but essentially when you see the color on top of it, that's it growing, meaning that it's alive and reproducing whatever, and it either has the nutrients it needs or it's a wild type. Yes.
In that diagram, was class 1, class 2, class 3 correlating to the enzyme number?
Yes, it was.
Okay.
And so this is going to be your example of that original experiment. As I mentioned, we're going to use oxytrophs a lot throughout the next 5 weeks. You want to be really comfortable with understanding how they work. So just think if you have a certain type of oxytroph, you need that particular nutrient in order for it to grow, and just grasp onto that. Okay, so is that clear?
Yes.
Okay, cool. Next question.
I just didn't it was the one with like green and then green sweater like that means that other.
Oh yeah. Okay.
And I know that it was gonna be because all that picture, but I'm seeing I think.
And it can, yeah, and that's the place where people actually do get confused. So I'm gonna actually go ahead and rewrite the answer choices. So A, B, C, D. Everyone did really good with phenotype on that question. So this is basically, we knocked out LEU2 and replaced it with neomycin resistance. So now we have a LEU2 oxytroph with resistance. And basically, everyone realized that in B, it's gonna die. This one has, wait,
It was without.
was that without both?
It had, no leucine and no.
Okay, so then I want to clarify, because I think in lecture, we said that everything died, but if there's no what's called, no antibiotic, the wild type will grow. So here you have only things that didn't get transduced, and that's one concept that we'll have to kind of go over a little bit, is that when you do these transductions, they're not 100% efficient. So it's gonna be something really good to post on the message board. So probably a point that actually I will post it. I want to make sure everyone is clear that you do this doesn't mean every single cell gets it, which is why we have to do selection in the first place. So only the wild type, which is our LEU2, is going to live. And then the LEU2 delta dies. That's without neomycin. And without leucine. Okay, so is this answer clear?
So the wild type can synthesize leucine on its own.
Yes, exactly. That's why the wild type grows. Okay, so is D clarified?
Yes.
Okay, good. B was clear. Like everybody crushed that one. So then we were basically left with A and C. A had leucine and neomycin. And then C had no neomycin. Neomycin, but with leucine. And basically, here we were sure that everything was gonna live. And we also have some things that were gonna live. So for B, so for C, what is going to live? You have two options. There's the LU2 delta, and you have the wild type. What is going to live if there's no neomycin? Everything. So that's both LU2 delta and the wild type. And then here, what's gonna live?
This is the delta.
Yes, only things that have resistance to neomycin, and that's your transduced mutant, which is your LU2 delta. So the reason why C is wrong is because you won't be able to figure out which one is your actual.
It doesn't filter.
It doesn't filter, there's no filter, there's no selection. That's what we mean by marker. Yes.
So we can use that pattern for future questions, like if something came up on these that it's like we give antibiotic resistance.
Yes,
If we get stuck between this, just go towards the option that has resistance to that antibiotic.
yes, yes. So you will see questions like this. You 100% will see questions like this. I know because I wrote the exam. So what I want you to do is, like I said, take the time to go through the phenotypes. Ask yourself whether the mutant is going to live or whether the wild type is going to live. And then once you figure out which one it is, the whole goal is to select for the mutants.
So did you say like a good bit of approaches? It's like kind of like go through like one thing, the first part, like filter through like,
What's-
oh, this would live, this would die.
Yeah.
Then add the new acid, okay, then this only one would live.
Exactly, exactly.
Okay, fine.
So always take the time to break down the prompt. Like, okay, what does this mean? So LU2 means it means the leucine, right? Okay, that's the wild type. I mean, LU2 means enzyme for leucine. And then since it's the wild type, it's fine and it can make it on its own. And I know it's a knock it out. I can't make leucine, you have to add it. So you want to be clear that you understand the scenario before you get to the question. Don't just like go straight to the answers. So a lot of students, especially when they're not used to like this type of application course, will go straight to the question, or they'll read the prompt and be like, why is there so much information in this prompt? Before it's multiple choice.
Yeah.
And there's information because you're synthesizing different pieces of information, because it's not just recall. So you notice there's a lot of different concepts you kind of have to put together in order to answer the question. But once you get the concept, you just work through it and it's actually relatively straightforward.
Hypothetically, like if it was, this is on exam, right? When we be told like what LU2.
Yes.
LU2 is. the one
LU2
that
is,
can-
yeah, produces a
Okay.
protein or the enzyme that synthesizes leucine.
Right, so you would like have to like.
You'll have to
know
memorize
that.
it.
Okay.
Yeah.
Thank you.
So yeah, you'll get pretty, you'll get comfortable with like LU. There's a couple others that are really common, so you get comfortable with them, but on exams, they will be defined. You're not gonna expect you to memorize it. On top of that, your exams are also open note.
Oh, yeah.
Yeah.
Thank you.
Okay,
You were saying that knockout is not 100% efficient, so it's when you do selection prior to this fact.
yes, that's what selection is for. And so it's actually one thing I'm gonna put something on the discussion board, that the selection process, the whole point is because this is not 100% efficient. And we'll talk more about like efficiency, I think in a couple of weeks, but you do need to know this concept to apply it yourself to get the questions right. Okay, next question.
So, we have like the same question, and when we were talking about the, melting point of DNA or DNA mutation, one of the questions was that how the increasing concentration of salt would lead to, increase of the, melting point. So my question is, I understand that the salt has this positive part ion that like interacts with the magnesium part on the phosphate and causes this increase. But you also mentioned that like, this like, could also like considering that like different circumstances, it would be different answers, so like, could you please elaborate on that?
Yeah, so I will elaborate, but the first thing I'm gonna say, no, you will not be quizzed on the impact of salt on melting temperature. Because of the reason that is so dependent on situation. The pH can make this effect of the salt different, the temperature can make the effect of the salt different, and other salts that are present can make the effect of the salt different, the type of salt can change it. So it's different depending on scenario. But the take home message that I want you to have, so say if you go work in another laboratory or you're working with DNA at all, is just to understand that the positive charge on the salt can actually interact with. Wait, we're still in the middle. Oh gosh, paint draw structures will interact phosphate. will interact with the negative charge on the phosphates. And then this actually helps to stabilize the DNA by repulsing water over here. So it's like a repulsion factor. And then so this drives the two DNA strands together and increases stability. So if you increase the stability, you usually increase the melting T.
So like the ion is kind of not surrounded the DNA.
Yes.
Oh, so, for the salt, the cation is acting more on the water than the salt like that backbone.
So the cation is acting on the backbone, but the effect of this is that it's going to change the electrostatic forces and you can repulse the water.
Okay, it's kind of like a shell.
Yeah, it's like a shell. That's a really good way to describe it. So remember, not an exam, but a good concept to know. Okay. Yes.
So I was reading on the video that we did in class, and it's about. Like literally, like shooting happens at the.
Oh, yeah, let's break that down. That one was,
Yeah, that's like the page 46.
Right,
That's the
yeah.
little lost on it, honestly.
okay, let's get unlost. It was orange. Okay, like I have the issue with like writing and spelling stuff, and don't, no, I'm so serious, it's really bad, I don't know why, last year everyone got really mad about my typos. So like, I'm getting my whole typo game up to get everything clean, like my TAs read everything, fix all the typos, don't make the undergrads angry at me. Okay, so. No, but I think it's like when you're stressed about the exam, other things can just stress you out, push you over there.
Yeah.
Right, so like, okay, let's,
It's like that on the final.
yeah.
Like, why is there no S on this?
I can't focus, okay. So you have these different enzymes. The enzyme essentially takes precursors. What's the first precursor for this
It
metabolic
was-
pathway?
Oh, it just said precursor,
Precursor,
and then,
yeah.
Oh, the pre,
and then it's
oh
a
my gosh.
foreign.
Yeah, so-
Organized.
Citrulline. Like I'm so used to actual,
It just
the
says
enzymes.
precursor.
A metabolic precursor that is unnamed. So essentially this enzyme will convert the precursor. Come join us, come join. It will convert the precursor to ornithine. Ornithine gets converted by ARCH2, this enzyme, to citrulline. Citrulline gets converted by ARCH3 to arginine. And then, so if you're thinking about an actual metabolic pathway, what the cell actually needs to live is whatever's at the end of the pathway. These are just the steps to get to the pathway. Yeah, I'm trying to think what you're getting. Like, you got the metabolic pathways and
Yes.
yeah. So it's like that. Think about the end product and the things that's necessary for them to live.
Mhm.
When we think about oxytroph, we're thinking of what is that last final metabolite that they need. So for example, if we block any enzyme in this pathway, we would call it an arginine oxytroph. Arginine. Because it can't live unless it has,
synthesize.
exactly. But depending on where you have the mutation, you can actually rescue it at different points. So if this enzyme isn't working, you can give it ornithine, citrulline, and arginine, any of them, and it'll still live because it has all the downstream enzymes.
It's like there's like a roadblock.
It's like roadblock, yeah.
Precursor and everything else.
Yes,
So
yeah.
You're not blocking like the ornithine, the citrulline, and the arginine. You're
Yeah,
blocking
and
that.
enzyme, that's what we're losing. So,
Thanks.
Takes a shortcut.
yes, exactly. So think about the oxytroph, that whole concept is always an enzyme. It's a very like specific set of mutations or like definition for types of mutations that you get. There are mutations in an enzyme that creates a substrate that is necessary for life for the organism.
Oh, that's sick.
So if the
Yeah.
mutation were to be at like 2, it would be as if the roadblock was at between ornithine and citrulline, right?
Exactly.
So anything, so you could give it citrulline or arginine,
Yes,
it would be okay, but
Exactly.
anything before that would not work.
I see.
perfect.
Okay.
Does that make sense? Is that clear?
That's crystal.
Yeah. Yay. That's something like this.
Because it can't go from ornithine to citrulline, even if it converted the precursor to ornithine,
Ornithine?
it can't keep going.
You
So
need
you
citrulline
can't-
or arginine.
So you need to add that. Like that.
It would-
add
If
citrulline.
you add arginine, would it work, or you would just. Arginine will always work.
Yeah. Good for you. That's the end product. That's what it wants. Like you're trying to get the cell to arginine.
Yeah, that
Okay.
makes a lot of sense. So basically, like TLDR, if you block anything before the name of like the
Wherever it
base
is.
molecule,
Yeah.
then you can't use them. There you go.
What does TLDR mean?
Too long to interview.
Oh my God, I'm old. Okay. Okay, next question.
I just had a few more going back to the previous lecture.
Cool.
Question one, just a few to kind of clarify over. So with a question regarding, talking about tyrosine and uracil, so you were saying that that's not a good determination used when you want to compare DNA versus RNA, because tyrosine could be found in RNA, and sometimes in uracil could be found in DNA sometimes, but we really need to be looking at is the 2' OH or 2' carbon, that's the main thing.
Yeah, so I'm gonna reiterate that. So that, of course, there are different sets of bases that are usually in DNA and usually in RNA. If I put a question on an exam, you will have them in your worksheet for discussion section, and I'm gonna ask you, which one is DNA, which one is RNA, or is it DNA, is it RNA? The only thing you should be looking for is to determine whether or not there is a hydrogen at the 2' position or a hydroxyl at the 2' position. So just think of the name. I'm looking for deoxyribonucleic acid, or I'm just looking for ribonucleic acid.
So just disregard the U versus.
Exactly.
Okay,
Freaking ignore it.
that's gonna be kind of hard, because I feel like in all of our classes.
And they're like, oh, yours. And that's something that you look at, but there's plenty of scenarios where yours still gets incorporated into DNA. Usually what happens, DNA is like, oh crap, I'm supposed to have that, and then you'll go back and do a repair and cut it out and put something else in. But sometimes it doesn't, and then you end up having to do, or that's how you get mutations. Like you put a U in on an accident, then try to fix the other side and cut something else off and replace it. Watch us talk about DNA repair, I think when week 5, yeah, when week 5. But uracil can get incorporated into DNA, so that's not what is defining what the DNA is. Or something can get mutated by like UV whatever induction to uracil, and then that has to get fixed.
And then when you were talking about the importance of the Watson and Crick model, just want to clarify one more time. So to make sure that it can contain information to dictate a cell's components was basically because it can do base pairing template for a new daughter strand. That's kind of main thing. And then when it can be easily duplicated to pass on during cell division, basically it said it's a linear polymer structure and it's anti-parallel.
Yes.
Anything else to add to that?
And the fact that you have enough base pairs to build a code that can code for actual proteins.
Is that for dictating cell components?
Yes.
So you have enough base pairs to dictate, okay. So enough BP to dictate the creation of proteins.
Oh, I'm prior topic of typos, there was a typo on. Yeah, you said cyanide for one of the nitrogen spaces.
Oh my God. You're very right. Okay, so I will go and I will fix the reading guide.
It's
Typo.
understood, it's like, you don't, it doesn't like.
I'm setting a new standard for myself this year. This is the year of the dragon, and I'm a dragon, so like, I'll get the typos right. Thank you for pointing it out.
I was reading, I was like, dang, they changed it, I guess science movement, but. I had a question for number 2,
Okay.
from the reading guide, it was, week 1, question 2 of the virus, dsDNA genome is unable to replicate itself in a host cell. What possible steps within the central document would be not functioning within the host cell? Do you just say RNA? I was thinking it was like a, what did I say actually? I said it was possible that there was like a mutation with either tRNA or rRNA, so like the ribosome itself wasn't like assembled correctly, or the protein or the tRNA wasn't translated properly. I was hoping to get some clarification on that.
Okay, so central dogma, we're reading DNA, RNA to proteins. So we have a virus that actually has double-stranded DNA, which means we're starting here. And then we're saying that it can't replicate in the cell, which means there's going to be some kind of issue or block probably here. So there's an issue with transcription or translation. And so the question is asking, is there an outcome there that is basically something wrong with transcription or translation? And any of those could be something that went wrong.
So it could just be like any of those steps?
Yes.
Pretty much, okay.
And so it's one of those questions that's trying to get you to think through the process and the flow of information. And you'll actually get more information on like transcription translation starting in week 6.
So but you don't have to know it in specific, right?
Right.
That was
So
the first one to think of.
Yeah.
The thing that would happen. I was
no,
like,
you gotta think about
no, this doesn't work.
So we really are focused on core concepts and applying them. And then for the reading guide is to help you think through the material and to start comprehending and remembering and understanding. So there's something about when you just first read something where it doesn't stick in your head. The reading guide helps some of those key things to stick in your head. And then what we go over in class, things I emphasize, the core problems, what you have on the worksheets, those are the things I want you to go back and study and work on and practice. That's what you'll see on the exam.
So really all the parts that go around.
Yes, yes.
Just be someone.
Yeah, so remember, the reading guide questions are to help you digest the reading and to really like commit some of it to memory without just like sitting there trying to do rote memorization. It's like memory that is based off of actively using the material, and it'll stick with you longer. And you'll go years later. So like the LAs, they literally came, I gave them the worksheet with your questions, examples from last year. They remembered like 95% of it. Like you will remember this stuff if you actively use it. But then once you get past the reading, like don't even worry about the reading questions. Like you don't need them, move on to the next thing. Okay, is that clear? Okay, who's next? We started zigzagging. Any questions? The newcomers? Well, the people who actually came on time and not 30 minutes early. Maybe we should shift office hours back 30 minutes. That kind of makes sense. It's like after class. It's right after class.
Yeah, I just like came in like.
If I had them like, want to get Starbucks or their brains are tired, but we'll just go right after. It's like 10 minutes. No, you were right.
It is fine.
No, I was about to come in and go to my office, like, I'm gonna get something to drink. I still people like outside, like, I don't know, outside. And that doesn't make sense to just sit here for 30 minutes. I'm like, no, we'll just start going for questions.
I'm sure we would have sat if you wanted to go take a break.
Oh, no.
Like, take the entire time.
Yeah.
I could have done a lot, I could have done a lot.
Oh, I don't want to. Okay, next question. We'll make sure everything is clear to you.
We're gonna go over this later. right?
Yes, so telomerase, you do not need 99% sure for first exam. Like it's not gonna, yeah, you don't need it first exam. And so I want you, the point of that is to really think about how many little components there are in like DNA, transcription, and translation that we don't necessarily know yet. And then we're going to go over these mechanisms in a deep dive. We're going to touch on the telomerase more for exam 2, and you'll see that in lectures 4, 5, 6, 7, 8, 4, 5, 6, 7, 8. I can count, I promise. You know, math is not my strong suit. Okay, hello. We're just going around. Any questions you have about the lecture, something that wasn't clear? We started early just because we did. And I think we'll actually probably starting at 3:30 in the future. I'll send an email out. And then it also gives us more time. Because like by the time I'm done, like what am I gonna do for 30 minutes end of the day? Like I gotta go home and get this baby. It's thing. Okay, next question. We'll go back and forth.
Okay. I was hoping you could elaborate on Oswald and Griffith's experiment. Why didn't the killed skinny bacterial strain back in the mouse like that?
It was completely dead.
Okay.
It was,
Yeah.
it was dead. There's nothing for it to go to. It was just all there.
How did
Okay.
it, how did
That's
it
my
affect the rough bacterial strain?
So let's just break this down. This is important to know, right? So when we say heat killed, I want to go through like what the actual impact is. I have a bacteria. Bacteria are going to have DNA. The DNA gets made from protein. The protein is what's going to have an effect on the mouse and like cause it to die. When you heat kill a bacteria, it can no longer make protein, which means it doesn't have the capacity to hurt the mouse. But what happens when you heat kill a bacteria, you basically have a shell and the genetic material is still there, because remember, DNA is hella stable. So then they put another bacteria, which this is a happy bacteria, not virulent, not gonna hurt the mouse. Bacteria have this really, really weird ability to take up exogenous material, especially genetic material, and essentially just gets the DNA that's hanging around. But because it has machinery to take that DNA, do transcription translation, it makes the bad proteins. So then when this goes into the mouse, it'll kill it. So basically we turn the good bacteria bad by giving it the bad bacteria's genetic information.
Is it all as one though? I was under like the assumption that it was like taking the entire like strain of bacteria as is. I know it's like circular. Like,
It's circular, yeah, it's taking this whole piece and boop, boop.
okay, so
This-
it's making it portions or anything? Like,
No, it just gets all the genetic material from that dead bacteria. And so this is the whole process of transformation, and they understood it happened. They really understand how. And what's funny is that transformation it's very easy and straightforward, but it can also be finicky. And different strains of bacteria take up different amounts of DNA, either more easily or more readily and less readily. But for the most part, it's, especially if you see a question about transformation, just assume that genetic material in the right conditions is going to be transferred to whatever is alive. So you heat kill it, the genetic material is left behind. And then what we've done is taken that framework, and then to make sure you understand the concept is inserted steps in between here. So forget that these two bacteria are together, right? We start off with the bad one. And they wanted to figure out whether the protein was important, whether the DNA was important, whether RNA was important. If they did a protease, it didn't make any difference. The DNA still went over there. If they used a nuclease that cut RNA, so only like a nuclease. Or let's see this one, make sure it's clear. So I want to make sure we're clear on the nomenclature. There's ribonuclease, a deoxyribonuclease, and then generic nuclease. Generic nuclease is going to cut everything. So I'll make sure that's clear so you don't get tripped up. Deoxyribonuclease, only DNA, ribonuclease, only RNA. And then so they tested each of those to figure out what was going to happen once it got taken up by a non-virulent bacteria. So it's just remembering the different components of that experiment and how each one works, how transformation works. And you can even like try to come up with hypotheticals and share it with your friends. Like, hey, in this situation, what do you think happens? And then when you start exercising yourself to come up with your own hypotheticals, you'll realize you really do understand the concept. Okay, any other questions about like the heat killed and why this works? Yes.
Could you just find that one more time? And then, do we have to know how exactly this transforms? But I just know like kind of like got into the DNA. Like the DNA.
Good question. So transformation is genetic information from one species or organism or cell getting into another species, organism, or cell. You do not need to know the specific mechanism by which the transformation happens. What's crazy is that when we first learned about transformation, they didn't even know how it happened. They were just sure it did. And now we understand different mechanisms. Sometimes it's like charge. There's even like ways where we force transformation to happen. And we'll talk a little bit about it in lecture week 4. Talk about it week 4. But you can like electrocute a bacteria and all of a sudden they just open holes and it's something what's around them, right? And then that's one way we get to that information into what you call it, into bacteria. Okay, is that clear? Okay, cool. So welcome everyone who came on time, even though it seems like you're late, you're not late, you're on time. Okay, other questions? We're just going around. If you have a question about anything in the lecture, in the reading, just bring it up now. We want to make sure we clarify it. Just to let you know, office hours is recorded. I will auto caption it using YouTube and then I will drop a link. Okay, yes.
I got a quick one going towards the Human Genome Project question that was brought up on the quiz. Talking about like Yak and Back libraries, configs, and like chocolate. I was hoping we would go over that in lecture today, but is that?
We did it.
So is that, not necessarily to really know specifically, what should we know for that board exam?
So
Human Genome Project?
it's not gonna be an exam. I forget what the Human Genome Project. So there's something to be said for exposure. So say you go into like a laboratory or another course and then you hear something come up. When you have we almost call it like recognition of like, oh, I know what that is. Then you kind of know where to go and get more information. So the reason why we're focusing not so much on a crap ton of content of memorizing, and instead of application is that once you have in-depth understanding of how to apply concepts, you can always look up the details. That's what researchers do every day. I stay on like Google, like, oh, look this up real quick, or let me grab my book, or let me pull this out. But it's harder to go back and be like, how does this actually work? Hello, come in.
I have a general question. Are we allowed to like start getting homework and ask clarification to
Yeah, hell yeah. Everything lectures 1 and 2.
Yeah.
Yeah, if you have, yeah, go ahead. Where's
I'm just like.
Yes.
Could you clarify about like selection methods? Because we were talking about video on as a selection method.
Yes.
I feel like we were moving a little faster.
We were, I was like, oh, we're running out of time. So yes, we were moving a little fast. We went over a little bit like just now, so I'm going to go ahead and reiterate. Because what we did miss in lecture was that the whole point of a selection marker is to make sure that we're choosing only the mutant that we created or that we want. So what I didn't talk about was that when you do the mutation, or you're trying to do the molecular combination, is never what we call 100% efficient. So let's start off. We have our original, let's just call it LEU2. And then we decide that we're going to take our vector or plasmid, we use this interchangeably. This has the NEOR on it. And then we have our DNA in here. We do the homologous recombination and we end up generating a cell that is LEU2 negative or delta, but then also now has neomycin resistance. That's what we created. But because it's not 100%, that means that we have some of this left over. We have these two in the mixture. And now we want to select for the one that we want by growing it in a condition that allows only this one to grow. So if we knock out LU2, it means that we have to give it leucine in order for it to grow. So the media has to have that. We have neomycin resistance. If we add a neomycin, the antibiotic, then this one will live because there's resistance. This one will die. So that's why the answer A was correct, because you have to be able to have only the mutant that you want live. If you don't have the neomycin there, this one will grow too, and you'll have a mix of both of these types of cells. Okay, you're welcome. Okay, next question. Yes.
So for the very acid, how was it? So can you in the example files behind you know, other bases, so we have a longer strand, let's say it was 100 bases, the first 5 match there, what's like a 5 prime in, and then 6 a match with 3 prime in, and then we create like a loop prime in outside of the map.
Yeah,
Okay.
so I'm gonna draw what's being described. We have, this is DNA, I'm just gonna say this DNA, this is my target gene that I want to identify. And we create a probe, the probe only matches here, it only matches here, and then you have this. Is this a good probe? No, it's not a good probe. And this is what would happen. What we want are probes that align completely with what we're looking for. Usually probes aren't designed to be super long, because we have to sequence them, and then there's also other opportunities for them to interact with other places in the genome. What we try to do with probes is design a medium-sized sequence, which do like 10 to 20 whatever base pairs, and look for areas in a specific gene that only that gene has. And that's how we design a really good probe. There's another concept that I didn't go over, so I probably shouldn't test on it. I don't know, we'll see. Maybe I'll drop it in the discussion board or in an announcement, be like, y'all need to learn this. But what's really interesting about hybrids, actually, no, we're gonna go over it. I will test on it. I will test on it. Yeah, I'll drop a note so people don't get by surprise. What's really weird about RNA is that if you have an RNA duplex, why can't I spell RNA duplex? This is actually super stable, very stable. I think you actually have some questions in your homework about it. If you have RNA to DNA, this is the next most stable. And then DNA duplex is the least stable. So because of that reason, people like to design RNA probes because they're very, what you call it? They make a really strong bond with the DNA. But then it was pros and cons when designing. One of the cons is that if you look at single molecules, okay, everyone's clear on this, yes? If you look at the single molecules, let's just take these two. Now, which one's more stable? DNA. Why?
Because it has. It doesn't have.
Already exposed.
Yes, I love it. Okay, yes, because the RNA has that OH, and this is OH. Yeah, I'm dyslexic. Okay. That's how everything back. That's where the typos come from. I've never diagnosed. Okay. This OH, when you have a slightly alkaline solution, is easily hydrolyzed. So you can basically break up the probe and it's just less stable for storage. So DNA is easier to store, doesn't get broken up, but then RNA makes really strong hybrids. So know the difference between those two concepts. Okay, any other questions? That was a good one.
Oh, follow-up.
Yes.
So you mentioned in your video regarding which to memorize all the types of RNAs. There's quite a few. You're supposed to know like mRNA, tRNA, and rRNA. Those mainly what else?
Yeah. So know the basics. They are going to come back up on the second half of this course. And like I said, I'm not trying to make Dr. Inley mad and be like, oh, I didn't tell him anything about any of the RNAs. I would like know the different categories. You won't need them for exam 1 or exam 2, but you will need them for exam 3.
So when it's like, would you use an RNA probe or a DNA probe? Would you say it's like situational?
It's situational. Yeah, and then so we would give you very clear situations of like, we need to make sure we have a probe that has really high affinity for the gene. Would you choose an RNA probe or DNA probe? Like RNA, yeah.
Okay, fine.
It's like, or we have to do this hybridization at this alkaline pH because of the type of, genetic sequence it is. Would you use an RNA probe or a DNA probe? Exactly. Okay,
Do we need to know like this ability of nitrogen mixes?
know the ranking. RNA duplex, RNA, DNA, DNA.
They mentioned one thing in the textbook, I didn't completely understand, it was, talking about probes, how you can use like smaller probes to identify like individual sequences, but like you do that. My understanding was like you use a bunch of different probes and then like you identify the one that, according to the duplex, hybridized with the part of DNA. So how are they filtering out like the DNA that doesn't, like how do they only get the double strand like when you come with like probes that don't hybridize to DNA?
So there's multiple ways to approach a, what you call, hybridization experiment. So when you have multiple probes in one, usually you're looking for like sequence homology. Because we have sequenced so many genomes now, it's actually very rare that the experiment is ever done. They just align the sequences and like, does anything match computationally? That's way easier. What they tend to do now is design a very specific probe. And I think in the homework, memory, yes, in the homework, you have some hybridization questions. They will use a specific probe, so they're only taking one probe, one sequence, with something to visualize it, and then incubating it with the host genome or chromosomes or whatever. The only other time you tend to see a mixture is when we do like the karyotyping, like I noticed. And what they do is add different ways to visualize, so different fluorophores, or like a tag sequence. So they give themselves a way to know which probe that they were putting in. Question, yeah. Oh, it just basically shows, it's a global picture of all the chromosomes. So you can tell if there's like an anomaly, if like a chromosome was cut in half, so you're missing like half of that, if you don't have all the pairs of chromosomes. So it's a very broad way to look at the overall genome of of organism.
It's not the same thing as fish, right?
They use fish to do the karyotyping. So fish is the way that they're visualizing it because they're using fluorescence. But you can basically do karyotyping. It doesn't have to be fluorescence. It could be some other tag. Yes, you have a question here?
No.
You have a question.
That's not what, yeah, about, there was a question on the reading guide that said, well, how did the induced genetic mutation from beetles and Tatum's experiments was under dogma? So what I wrote, just from my memory, to make sure I analyze it correctly. So prove that one gene DNA forms one enzyme or basically polypeptide, by finding that mutations via like optotrophies were mapped on the same chromosomal location as normal genes. So a mutant is caused by a single defective gene. And then basically saying what an autotroph is a mutant organism that requires a particular additional nutrient that the normal strain does not. Is there anything else specific with these beetles and potatoes that we should know?
No, that's the first thing they did. The first like some background knowledge.
Yeah.
So they would take these autotrophs and they tested them against all the different like nutrients that they think it might need, to, like rule out like whether it's a single enzyme or not. So the experiment workflow is very, very simple in the textbook. But really, they had a whole bunch of different samples, a whole bunch of different mutants, a whole bunch of different conditions with different individual media to test them all. And it's basically deduction, right? So you're checking everything off as you go until you arrive at, okay, this is the one mutant that it's only has issue with one enzymatic pathway and one particular nutrient. And then that's how they can.
So that's for the central dogma specifically, and just kind of prove that, yeah, it's DNA that's holding the genetic information.
So that experiment actually had nothing to do with central dogma.
Oh,
That experiment helped us to understand or help validate that one gene is related to one polypeptide or one protein or one enzyme. And this is the first like way of us getting at that we think it's one enzyme. Yes. Yeah. So model organism. Sometimes when you don't recognize something, you try to break down the actual words. So model, we're trying to model something that's complex, and usually it's humans. But in the laboratory, first of all, you can't just do stuff to humans. It's not ethical. We also take a long time to replicate. So if you want to look at our DNA, it takes forever. There's some cells in our body that might replicate, others don't. Also, our cells are very complex for eukaryotes, right? Or eukaryotic cells, all the different chromosomes. We have like two versions or two sets for all the chromosomes. So what people do with model organisms is use something that we call lower order, where they're more simple, they either divide more easily, they use or require less nutrients. So it's what we call a reductionist approach. You find what you want to, the question you want to ask, and you figure out, is there a simpler organism that I can use to ask that question? So they use mold to ask a question about DNA, right? People use so many different types of model organisms. Like in our lab, we use laboratory mice to model like complex human disease, or to model aging. Like we're not gonna wait till somebody turns 100 and figure out what happens to them. It takes too long, right? So you model it in a mouse. There's people on campus who use mosquitoes and genetically engineer them. We know so much what we know about humans because of stuff we learned in fruit flies. So model organisms have the power to teach us a lot about basic biology. And then we translate that later and test the mechanisms we find in the organism that we care about. Oh yeah, so haploid, what's cool about that, so remember I mentioned like humans, we have 2 copies of all of our chromosomes. So say, for example, I got a mutation, or I have a mutation in one gene in a chromosome. There's a possibility that my other chromosome has the wild type and it just, you know, takes over and I have a dominant allele. And then so you don't see the effect of that mutation. What was beneficial about being haploid is that there's only one version of every single gene. So if there's a mutation, you are going to get a phenotype. Yeah, you don't gotta worry about all those Punnett squares and the autosomal dominance and negative and recessives, none of that. Genetics wasn't for me. Okay, next question.
It was regarding the reading that said, this can briefly describe the different types of sequences within our genome. There was quite a few listed, like lines, signs, retrotransposons, transpons, SSRSs, and all introns. You kind of went into like a few of
A
them.
tiny bit of them,
And then,
yeah.
is this another one of those like callbacks? It's like, oh, just for our exposure?
Awareness, yes,
Awareness.
exposure.
But which specifically should we know? Is just basically intron, UTR, that.
Yeah, the ones that popped up on the slides.
Okay.
And they're gonna come back into play starting week 5, when you start going over transcription. So you want to know what an exon is. You need to know what an intron is.
Yeah.
You know, 3' UTR, you're gonna need to know them.
Okay.
So understand what they are, know that we're gonna come back to it.
Okay, awesome. Thank you.
Yes.
It's more of a hypothetical. And so we're able to knock out genes, so why do you not, like, are we able to knock out any of the gene in humans yet? Is there, CRISPR closest thing that we can show this in the ways, or what?
Yeah, we can 100% do it. All of the issues is not the ability to mutate or change genes. It's to mutate and change genes in the cell types that we need to, and doing it in the body and so it's more so like logistics issue as opposed to can it be done. And then after you get to logistics, a layer of safety. So, and then also because your carryouts are very, very complicated. So like if we're going to change a gene, what are all the bystander effects? What are the effects that are going to happen if we get rid of this gene? Something else bad is going to happen because this gene is gone. If there's something good that it was supposed to be doing that now we don't have it anymore. And then also the extra complication of the fact that, like, so we have 2 copies, 2 alleles of everything, and understanding the genetics of how any type of mutation works. Is it recessive? Is it dominant? Do you only need 1 allele or both alleles? How effective or efficient are we at actually getting rid of both alleles? In vitro, all of these techniques, especially CRISPR, work so well. Like CRISPR immune cells change genes, modify them all the time. The CRISPR screens. We're getting better at engineering cells that we can take out and put them back into bodies, but actually genetically engineering cells in the body is a little bit difficult. It's a cool question. Yeah, not an exam, but it's cool. Yes.
I have a question about outside forms.
Yeah.
When I asked for examples, I was wondering, it's like an example, but it's also inside and last reason.
Yes, exactly. So, and any organism that is missing the ability to make a nutrient that is essential for their growth, that is the oxygen. So it needs that nutrient to live, it can't make it. No problem, yes.
So the follow-up question, and hypothetical. And so, if hypothetically, when you screen the parents with their fertilization, and then you find out that you don't have it, let's say see that it is a recessive, are we able to not have been knowing hypothetically, assuming you have it?
100% hypothetically, yes, but not even like assuming we have the technology, we have the technology. Those things right now are ethical considerations. So that's why they're not done. Did y'all see the news article? I think I might bring that up later. There's this news article of this guy, I think he was in China, and then he like reversed a mutation that would actually cause like disease.
Yeah, and then he got into like.
And then he got, yeah,
Oh,
like,
God.
Twitter went crazy. I'm like, oh my gosh. But it's like, it was a good thing, but it opens up the avenues for like a lot of potential bad things. So we're like, so people just kind of stay away from it, even though we 100% could do it. Okay, any other questions? Yeah.
When I said the ball line on the highway, I was just wondering, what is the difference between RNA growth and being, something
Yeah, so that's what we're talking about. We're talking about the different hybrids. So basically, we're deciding whether or not we're going to use RNA to probe for a specific sequence in the genome or use DNA to probe for a sequence in the genome. And understanding the relationship and their stability either as a duplex or individually is important. So remember, as a duplex, RNA with RNA is the strongest, then RNA to DNA, then DNA to DNA. And then when they're individual, their stability is different. So DNA is more stable, single-stranded compared to RNA, single-stranded.
I think it's.
We'll go here. No, well, actually, no, there. Okay, your probe is always gonna be single-stranded. So remember, you want to form complements. So always gonna be a single-stranded probe. So the probe and the target, they hybridize, makes me double-stranded. So think about what that product is. So if I'm looking for something in the genome, that's DNA. If I have an RNA probe, then they come together, you have an RNA DNA hybrid. And then so you think about that product, is that more stable as opposed to like I made a DNA probe, now I have DNA hybrid. Yes, question here.
So, or so you said the RNA related, that's just so, this was just a question about the election. There's a slight audit of mine, true. It just says that it's like not true, that it's possible that I hybridizing RNA into another RNA is that? Is that like?
Can you read through the whole question?
Yeah, so it says like which of the following is true regarding nucleic acid? Saying we don't apply. The selection that is wrong, it's always possible.
Oh yeah, just come show me.
It's over reading all that.
Read me everything and let me comprehend. Okay, we're checking a reading quiz question. Okay. Choose all that apply. It is possible for visual regions of DNA and regular probe. That is true. It is possible for DNA to reduce hybrid. That is also true. And DNA have reached the spontaneous at room temperature. Yeah, they're all true.
Oh, it was just marked wrong. Okay, so was it just like a?
Oh, this very first one. This is not marked.
Yeah, so when I first took the quiz, I marked it, and it was like marked wrong. When I redid it, it was, so like, is that one?
Yeah, that was true.
Okay, so.
Yeah, I'll double check just to make sure I'm not post it. But yeah, that's true.
Thank you.
So on the homework, it has like the RNA DNA process we went over, but there's also an interface talking about the antibody complementary to the mutation or whatever.
Yeah.
What does that actually have to like do in relation to versus like DNA probing and RNA probing?
Nothing. If you know how hybridization works, you recognize there's no antibodies in the process,
Okay,
right?
okay.
Okay, good looking out.
Wait, so what's it? Like it doesn't like have
Uh-huh. Recognize antibodies have nothing to do with this whole process.
I'm like a little confused.
Was this classification, so those DNA grow from saying like one strand of DNA, like one?
Yes, so a probe is a single-stranded DNA or RNA.
Just one.
Just one.
It's not available.
No,
It's not available.
it's not gonna be a duplex.
Oh.
So a probe is a single strand by definition. Yes,
Why is RNA duplex more stable than everything else, even though RNA itself is less stable than duplex?
yeah, so for some reason, there's something about the actual like biochemical shape interaction it has based off of the pairings of, it's the uracil. Which side is it? Which is the other side, y'all? A, it's that U to A interaction. That is a little bit more stable. Okay, other questions? Yeah, that was one of those things I've like, check my notes. Yes,
One was this, I guess the reading, to kind of clarify. So it said, you choose two people in the world, and is it true that they were, true or false, that they would have about 80% genetic similarity? I'm not specifically in the reading, but from what I remember, it's like 99.9. it's false, because it should be 99.9% similar, because we only differ like 1 to 1,000 base pairs, something like that, but.
Yeah. So this actually goes back to like principles of Chargaff's rules. So within a species, basically all the DNA content, the ratio is essentially the same. So at a broad perspective, right? So our DNA for human is basically all the same with small differences in like SNPs or point mutations. But then genetic content is going to change from species to species. Yes.
No, right. And how does this sequence go to affect the observance graph? Will it change or is it always constant?
The sequence will affect it if you change the G to C versus the A to T content.
Okay.
So that's the only thing in the sequence that's actually going to change it. Yes.
What does, increasing the buffer concentration, what does the buffer actually do? In terms of, like, does it add, because it's like NA plus and CO minus, right? So what is adding the buffer or increasing the buffer concentration do with like the temperature at which the DNA begins to?
Okay, so that buffer one, that's one we actually spent time in that I said is very confusing. That will be at homework. I want you to learn it, but will not be on exam. So for the buffer concentration, they're saying the buffer concentration of the salts, right? So you're increasing the amount of positive ions and basically stabilizing the DNA because of the
So-
interactions with the
So it
phosphate.
would increase.
Yes.
Oh,
Yeah.
okay, got it.
Yes, here.
You see more just like, you know, kind of question, but.
You ask me chemistry? Okay, go ahead.
Yeah, if they're not in the funding space, and how they're considered hydrophobic, because they're going to nitrogens and then they have like a leading bond, and I'm assuming that how much we apply that, like. Okay.
Oh, man, so don't ask me chemistry. You're gonna go out there, put on the light belt. Yeah.
Like all the traits that it's obviously like there's hydrophobic, something like molecule, you think that it's.
Hydrophobic, yeah, you would think that, you know, like maybe it would like water a little bit, but they don't at all. Yeah, they don't like water. Just know, that one just memorize. Okay, I saw a question back here. Yes.
No, I. Oh,
Okay.
Could you provide, I remember, can you provide an example, so if we introduce the, you know, like, and now, does that make it less stable? Does it, I don't know what you say, it make it less stable?
Yes.
Okay. And what if you introduce it to like a salt that have, like, when out there, into a salt, so by it not changing, then you have the positive and negative, or like.
I mean, technically it shouldn't. It's actually, it's weird. It's oddly complicated and I actually can't give you all the mechanisms because I don't understand it myself, which is also why we don't test on it, because it doesn't necessarily make sense unless you go through the exact mechanism for every single individual salt. And like I said, you can salt DNA in and salt it out.
Right.
So it's highly context specific. I wish I could give you a better answer, but that's what I got for you.
Thank you.
Yes.
It's kind of like the same thing you just had, but, you also like, explain how like, pH, because you said if it's slightly basic, it can like help it, but then the belly, I don't know, like, in regards to like extreme conditions of basic and acidic, like which one decreasing and decreasing DNA. And then, could you also just go over again, the salt, like, how, there's a lot of salt and, you know, like salted out, but then it's just over salt, and then, like, why did that happen? Can you say that was something like more heavy?
Yeah, so just gonna touch on the general principle for the salt is that you increase the positive ion, it interacts with the phosphate backbone, therefore stabilizing it. It excludes water through electrostatic forces, basically pushing the DNA molecules together. So that's how the salt works. That's general principles. There are a lot of exceptions to the rule. For those reasons, you will not have a question on the exam, but I think you have it in the homework. Know the general principle. So for pH, what I want to do is go over pH on x-axis and then absorbance on the y-axis. We're going to call this neutral, 7, 4. This is going to be a little bit basic. It's going to be extreme basic. Little. And this is going to be a little bit acidic. This is going to be extreme acid. Okay, so what do you expect to happen to DNA in extreme acidity or extreme basicity? Is that a word? Acidity, it's a real word. It's going to denature it. If the DNA is denatured, are you going to have high or low absorption of at 260?
Hi.
Okay. Now, it's a little bit basic. What happens to DNA when it's slightly basic? Is it more stable, less stable? And why?
Because there's one acidic and one that I think that the request to have to possibly make the DNA more stable. And if it was the opposite, it would be the opposite.
Okay, so yep, we have basic, which means we have extra hydroxyl groups. We of course, we're stabilizing because we're what you call it? For the hydrogen bonds, you basically want to be able to have hydrogen available, right? And then this would take away those hydrogens. So it's destabilizes exactly like you said.
Okay.
So it's clear to everybody, yes? So if it's not super stable, what do you think the absorbance is gonna be about 260?
Or lower than the extremes, but higher than if it were a little less.
Okay, so we're saying we're probably about like right here, yeah?
Yeah.
Okay, so if we're at page 7.4, what's going on with DNA? It's at a happy place. Yeah, it's probably normal, like nicely double stranded, all duplex. What do we expect to happen when you have duplex DNA? How's the helix? Low absorbance. Okay,
So absorbance is only looking for single stranded DNA?
versus, yep.
Okay.
So remember that whole like hypochromic and hyperchromic effect? The more compacted DNA is, the less it's going to absorb. The more like spread and free it is, the more it's going to absorb. Okay, so now we're to pH here.
Mhm.
It is a little bit acidic. What is going to happen if it's a little bit acidic?
Does it stay the same as the lower, because you have more hydrogen?
More.
So we made it
It's
even
more
more
stable.
stable and more like compact.
Right.
So we're probably like Harris. And then we said an extreme. We just totally destroyed it, right?
Yes. Right.
Up that, okay. So, and so if you saw a graph like this, and I said, what is the x-axis based off of this being absorbance of DNA? You can tell that's pH, right? If I drew this, that's temperature. So know how changing these interact, like you will 100% get one of these as an example. Give me the x-axis.
How can you lower absorbance if like, the lower acidity? How do you get like even less single stranded DNA than exactly?
So it's extra compaction because it's stable, so you're lowering the melting temperature.
Okay.
So the amount of compaction, it's not gonna go down a whole bunch,
It's
but
just
it'll
like-
go by about equal, or like slightly less.
Is it like marginal? Is it like this?
It's more, yeah, it's like negligent. So you're definitely not going to, yeah. And then, yeah, I'm usually pretty sure, like, I try really hard to make sure that answers are clear, and just FYI, if something isn't clear, if there's actually something wrong with X on the exam, like, feel free to be like, hey, I think that was wrong, but it's not the XYZ, these are my reasons. And then everyone will love you because they'll get all points back for the whole class. So, and so if you have a legit reason for like, I think this is correct for this reason, based off the way the question was written, just let me know. Okay, other questions here?
And now we're hypothetical.
Hypotheticals, let's go. No, honestly, this is the way I want you to think. Start thinking in hypotheticals and it'll help you process and you'll be like, oh, this test is so easy. That's how you're gonna feel.
So we established that if we did a development of DNA and pop media and then two single stranded DNA. So what we're is we say single stranded DNA in a very important and your ones will stay separate.
What do you think?
Stay separate. If you place them in a real column, very like a person.
What do you think? I want to hear what hypothetical is. I have an answer. What do you think?
I think it's going to stay separate.
So they're still going to anneal, but it's gonna be slow because it's so cold. Exactly. Yeah, I like the hypothetical. That's cool. Should turn into a question. You'd be like, I wrote this question. Oh my God. You'd be like, I wrote this question. Okay. Yes.
Could you explain that also?
Yes. No. Hyper. Chromic. Chromic meaning color. Okay. And then, so think of a DNA backbone, this is so ugly, base pairs. Okay, so if I was looking at super, oh my gosh, I'm just breaking things. That's super compact. And then I'm gonna shine some light on it. And I want to absorb this light. How much harder is it for light to get into this molecule to get absorbed? Yes. If I have everything spread out and I have a couple of my bases flipped in and out because it's single stranded and you know, a little bit free, how much easier is the light to get absorbed into this molecule? Yes, so even though both of these have the same structure for those aromatic rings and absorb at the same exact wavelength, it's easier for this to absorb, so that hyperchromic effect, and more difficult for these to absorb, so hyperchromic effect. Yes,
Is there one factor that's more impactful on your absorbance studies, like this huge one factor that factors for the starts or you about this?
so don't think of the effect of pH on the hyper or hypochromic. It's the effect of pH on the ability of the DNA to either denature or renature. Like what position is the DNA in? And then from there, they're like, okay, how high or low do I think the absorbance would be?
Got it.
Yes.
Question, I don't know if I should hold off for next, but it was on the lecture 3, lecture 4 readings, I don't know.
You should probably hold off because I record it and then I say, this is office hours for lecture 1, lecture 2.
No problem.
It's cool. Maybe catch it like right at the end if you want to go ahead now. Like when we're done. Okay, any other questions? Yes.
The Hershey Chase experiment, and it's not found in the relationship or something like that.
Okay, cool. Hershey Chase. Hopefully, majority of us are like starting to grasp it, and we know it is actually one of my favorite experiments. Let's start off with why in the world was Hershey Chase done in the first place? Can anybody tell me?
To determine what actually carried.
Yes.
Yes. So what carries genetic information? It's so weird because like I think about what we know now, I'm like, how do we not know? But like, but they didn't know. So their idea was they were going to utilize a model system in order to ask and answer this question. They decided to use the system of what we call bacteriophages. Because this is a virus that infects bacteria and uses the bacteria to replicate. So bacteria use it to grow. The viral particles are also relatively easy. So the other bacteria is basically two different things. You have a protein coat, and then you have nucleic acid. And they want to know which of these two things is taking the information, the genetic information, putting it into bacteria and allowing it to replicate. So what they decided to do was label either the protein coat or the nucleic acid. So then I ended up giving them two separate samples. So I'm just going to erase this. This one protein was labeled and it's labeled with radioactive sulfur, a radio-labeled sulfur, and then this one had the nucleic acid radio-labeled with phosphorus. This works because phosphorus is only found in nucleic acid, sulfur is only found in the protein. So it has to be one of these two. So after they took these radio-labeled viruses, they infected bacteria, meaning that whatever's genetic information should get passed to the bacteria. Then they put it in a blender. So zit, 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 zit. And what was so cool about this very simplistic approach, that the blender was medium to slightly harsh and caused a separation of the virus from the bacteria. So now they can do what we call a centrifugation step. And to determine whether or not they found label in the bacteria or not. So for the bacteria, it pelleted because it's larger than the virus and it's heavy. But what happened is that we found a label only in the supernatant. So sulfur, 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 sulfur. Then over here, when we labeled the nucleic acid, we found a label in the pellet. Phosphorus, 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 phosphorus. And they're able to detect this by basically using a Geiger counter and determining is there, are we getting like beep, 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 beeps here or getting in the pellet. When they found it in the pellet for the phosphorus labeled nucleic acid, they knew that it had to be nucleic acid that was the carrier of genetic information.
Because that was what was found actually.
In the bacteria.
And the sulfur was only outside.
Only outside in the supernatant. So it never got transferred to the bacteria at all.
So in like the head that really said in class, or at least that the protein was actually the genetic carrier, That's just like in the hypothetical, in the sense that the protein would somehow enter the bacteria.
Well, somehow end up in the bacteria.
Somehow.
Yeah, so that's the hypothetical. Yes, question here. Project Griffin is like, I'm shooting up the mouse with the bacteria that we mix together, right? Okay, and then also Avery, give me a quick reminder of what they did again? Okay, those are sequential. So 100% related, the concepts are together. So the very first ones was essentially seeing that I could kill the mouse with the genetic information of the bad or lethal bacteria that got transformed into the non-virulent strain. And then the next researchers went a step further and was like, let's figure out exactly what material it is. They used like the protease and the ribonuclease, the nuclease, and figured out that it has to be the DNA that gets transferred. Yes.
Distinguishing between the Avery experiment and she takes it something like kind of like saying DNA and everything.
Yeah, so one thing that's really cool about science, or actually it can be cool but not cool at the same time, but when something new is discovered, usually we want a preponderance of evidence, right? So multiple different experiments that answer the same question and get the same answer. And they were like, yep, I think that's true. So that's what's happening at this time, is that you have contemporary scientists trying to find ways to address this question using different model systems. They arrive at the same answer, that DNA, nucleic acid must be genetic information. Okay, any other? Yes.
For the PGK rhesus, chemistry is by is the sulfur or the phosphate doesn't get into the nuclear, is why the DNA will carry the genetic information.
Yes, so I want you to know every single part of this experiment and what every single piece is for. Like, know this experiment inside and out.
So the total was asking them that. Is why
Yeah, so the pellet, when we labeled with phosphorus, which labels the nucleic acid, that was found in the pellet, which is how we know that that was the genetic information getting transferred. So yeah, go through, like, review this experiment, every single piece. Like, why the blender? What's it for? What did it separate? What did these label? Know it. Yes.
Wait, why was it found in the pellet? It was just like, integrated itself in did it infect the bacteria?
Yeah, it infected the bacteria.
And then
So-
the sulfur didn't affect the therapy.
Yes.
Oh.
So protein from the virus did not get inside the bacteria. Nucleic acid from the virus got into the bacteria. Any other questions? Okay, so we're at 4:45. So for those of you who've been here an hour, 15 minutes, like, you're not prisoners. So you want to go, feel free to go. Members recorded. I'll post on Canvas tonight. Any other questions? Okay, if not, go to discussion next week. If you're Monday, go to discussion. Like, you will thank yourselves for going. And then I'll see you all on Tuesday. Okay, bye, everyone.